I haven't bitten anybody yet.
Keep it that way then.
Yeah.
Is our exam gonna be in this room?
Yes, right here. So, what you might want to do, I don't know that do I need to go over each one or can you just look at this? And I would probably try to write down some corrections. Just so that, one of the things that would be advisable to do would be to take all your exams, and even though the questions may not be taken exactly from the other exams, a lot of the subject matter would be similar. I would reread your exams and make sure you get the right answers programmed. Can you see that or not? Do you want me to read them?
Sure.
I mean, I can do that. Okay, 1 is anatomy, 2 dorsal, 3 negative, 4 connective, 5 false, 6, I wasn't trying to, but I reversed what I normally ask. I asked you which ones are water soluble, and it's B and C. Adding is fat soluble. 7 glucose or monosaccharides, amino acids for 8, 9 proteins, 10 is the alimentary canal, is also the food tube, 11 pyloric, 12 pancreas, 13 small intestine, duodenum, 14 protein, 15 pharynx, 16 colon, large intestine, 17 is right, 18 skeletal, 19 calcium, 20 false, 21 is positive, 22 action potential, 23 SA node, pacemaker, 24 arteries, 25 pulmonary, 26 right. You don't have enough light to see, do you? Is that better? Okay, 26 was right, 27 vena cava, 28 aorta, 29 left ventricle, most muscular pumping chamber. Okay, 30 is red blood cells or erythrocytes. 31 alveoli. 32 trachea. 33 true. 34 bronchitis. 35 epiglottis. 36 nephrons. 37 true. 38 glomerulus. 39 Bowman's capsule. 40 is true. 41 is a heart attack or a myocardial infarction. It's the same thing. 42 hormones. 43 pancreas. 44 thyroid. 45 pituitary. 46 is false. 47 is iodine. 48 hypothalamus. 49 is either FSH or LH. 50, either epinephrine or norepinephrine or adrenaline or noradrenaline. 51, I was looking for the unbroken skin, outer membranes. 52, lymphatic. 53 is the innate and the adaptive immune systems. 54 B cells, 55 antibodies, 56 helper T, 57 thymus, not thyroid, but thymus, 58 true, 59 vulva, 6 the clitoris, and 61 of either the range of 12 to 16 or the 14th day. Let me just remind you that the reason why with the day 1 of the female uterine cycle is the first day of the menstrual flow. And so around 12 to 16 is when ovulation of the egg occurs. And so as soon as it's ovulated in that upper 1/3 of the oviduct is when it's the most fertile time. So ovulation is going to occur right around that time. Okay, 62 in terms of the epididymis was to store and mature sperm. 63 seminal vesicles. 64 false. 65 males. 66 progesterone is really the hormone that most helps prepare the uterine lining for receipt of a baby. Okay, 67 the central nervous system is the brain and spinal cord. 68 is the parasympathetic, which is the brake, whereas accelerator is the sympathetic system. And then the answer is 69 is 1, 70 was 8, 71 was 1, 72 was 3, 73 was 7, 74 was 8, and 75 was 5. If you want to double check the arithmetic on the test, like how many you missed at the bottom of each of these 3 columns. And I usually put that at the upper left, minus however, and then if you got up to +3 or plus whatever, I subtracted that from the number wrong and then came up with a net number times 2. There were 75 questions, so 75 times 2 makes it 150. So then I subtracted it from 150. So make sure that the arithmetic is correct. Anybody have any issues there? I'm sorry, I can't hear you.
I have one that you marked wrong that wasn't wrong.
Okay. I'm bringing up your Let's do the light on. Let's see. I needed the helper because there's a type of T cells. Oh, you know what it is? I'll mark the wrong one. This one's wrong.
Oh, okay.
I'm sorry,
That's fine.
because that's supposed to be vulva.
Right.
I'm sorry.
That's okay. Thank you.
Any questions about this test?
Yeah.
Yes.
So on number 69, it says to identify the dendrites in the image. So which one would that be?
Is root line lucky number 1?
To be number 1?
Yeah,
Okay.
yes, number 1. It's the beginning, the lead end of the neuron, that's a neuron.
Sure.
So about using the axon. One time I didn't bring it. Any other questions about the exam? Okay, which brings us to the final exam. Our other purpose today is to try to get you primed a little bit. All right, let's put this up. My allergies. That's it. Okay, so topics for the final exam. Let me turn this light on. We there's taxonomy questions, which is the science classification. Who is the father? We said the father of the original kingdom, phylum, class, order, family, genus. Things play chess on 5 spaces. Kingdom, phylum, class, order, family, genus, species. Linnaeus is known as the father of taxonomy. Kingdom, phylum, class, order, family, genus, species. There's also a question on the final exam that, well, there are some multiple choice, but you still got quite a few fill in the answers. Type questions. Yes. 90. So you'll have 2 hours. I guess I'll probably give you about 20 minutes with your notebooks. So double the time. So if the kingdom is really large, isn't it? Because an example of a kingdom would be animal kingdom. If I tell you I'm thinking of an organism that is animal kingdom, do you know what I'm thinking of? But if you really knew the genus and species of it, you would know it, right? So the higher up this grouping, the more organisms that are included in the group, and it says the least about it. There's a question that asks you to rank which has the most individuals in it. So between class and order, which one of these would have the most individuals in it?
Class.
So class would have the most. So you have to remember it goes that way. If I'm asking you a question that says what identifies the most specific, it would be this direction. Okay, how to write a scientific name. I did ask you to choose, I think, which one is correct. And I'm not sure if I used our scientific name or not. Remember the 3 points to how to write it. The genus, this is the genus name. This is the species name. Capital letter on the genus name. Small letter on the species name. And the third thing is underlined. Or italicized, but I usually use underline. I got some cheat sheets. Okay. Okay, well, while we're doing this, since Linnaeus' time, we've also now identified something bigger than the kingdom. So now it's a B up here. Okay. And what are the 2 prokaryotic domains? Yes. Very good, thank you. Bacteria, archaea, and then the third domain is eukaryotic, and it's called eukaryotic. Right? Thank you, very good. Let's keep it safe. What does it mean to be prokaryotic? What is it the prokaryotic cells don't have that eukaryotic cells have? There's one big structure in the middle. The nucleus, very good. The nucleus is missing. So there's no nucleus in a prokaryotic cell. It just has a circular DNA that's not bound by a nuclear membrane. Okay, so no nucleus. And very simple cells. Binary fission, division, just very simple cells, but bacteria and archaea. Now, let's talk viruses. Where are they in this taxonomy? That could be a trick question. They're not.
They're non-living.
But we studied them because they cause so much damage.
Mm.
Well, how did we describe viruses? Their lifestyle?
Cellular. Parasites.
This is a bacteriophage, this is a virus. It has an outer protein coat called capsid.
DNA and RNA.
It has a nucleic acid core, and it is either DNA or RNA. So there are RNA viruses and there are DNA viruses. They have no cellular structure. You might want to review the characteristics of all living things. So we really don't put them in the taxonomic scheme yet because we really don't, do we consider them living
Some people do.
a real life form? No, they're just, they say, basically they say, we say they're entities. Okay, it's kind of it's not a real cellular, it has no cellular structure. They're entities, they're obligate intracellular parasites, which means they use the cell machinery of the host cell to make more viruses. There are two cycles that they can become involved in. Lysogenic and lytic. Lysogenic is sort of the sleeper. It doesn't do immediate damage. It sort of lays there dormant. And then all of a sudden, it will eventually turn probably lytic, which is the most virulent and infectious, most damaging. It will cause lysis to occur in the host cell, which means it'll cause the cell machinery of the host cell to make all these parts of the viruses, and then it will burst the cell membrane of the host cell and will release all these little viruses. And lysis means to burst, lytic. Okay, there is also we barely mentioned this, but unfortunately it appears on final exam, is that there are entities called prions that are sort of like a virus, but they only have the protein part. They don't have the nucleic acid part. And these things of becoming, they're induced, there's a disease called mad cow disease that is caused by prions, which are proteins that are folded weirdly, and they actually become pathogenic disease causing. So some of the sound at least vaguely familiar. Okay. The word virulent means highly infectious, or it's something that's more virulent is more damaging. Okay, now we need to talk bacteria. Remember the three shapes? Coccus is round, bacillus is broad, and spiral or comma. Now, remember the arrangements? So shape, and arrangements when they divide. You can have diplo, you can have, what is this called when they're in a chain? Think of strep throat. Strep. Strepto. Okay, so this is strep. And then if there are clusters of grapes, it's called what? Other than strep, what's the other famous? Staph, thank you. S-T-A-P-H. So staph is clustered, strep is in a chain. Now, if I ask you, be careful to read the question because I'm either going to say, like if I pick strep, I could either say to describe the shape and arrangement of something that is Streptobacillus, then that would be like this. A chain, but a broad shape. Or if it's Streptococcus, it would be a chain of round. So another structure, some bacteria that actually are the most resistant forms of life known. Some of these bacteria, like in the genus Clostridium, Clostridium. Clostridium's an algae, I'm sorry. Clostridium. Like tetani, botulinum. This is the genus, this is the species. What is it? They can go dormant if conditions are not favorable. It's something that forms inside the cell, so that's key to the first part of the name. And it's how we have to use like an autoclave or something very, very serious to kill them, because boiling water will not kill them. What are those structures that some can form? Anybody know? Some bacteria are endospore formers, and that's why we have to use the autoclave and other very serious methods of sterilization. Okay. So I think we can do pretty well on this type of information. A lot of this will be near the beginning of your exam. So what would be next would be the protus, some things about the protus. Because remember in eukarya, domain eukarya, you have protista, fungi, plants, and animals. So we know this is the botany stuff, and we are going to have some botany information. And the animals, there will be some zoology questions. And this is mycology. There will be some fungi questions, and there will be a couple of protus questions. Remember these words. Plankton, keyword. And zo would refer to animal-like, and phyto refers to plant-like. But these are protists. Where are they found? In what kind of habitat?
Warm,
In what?
like high temperature.
Somewhat warm, but not necessarily. Wait,
Is this what you?
are we talking? Okay, let's just narrow it down. Water or terrestrial? Water.
Oh, okay, I was thinking.
Yeah. So the phytoplankton is all that algae type stuff, some microscopic that you can't see, that are actually in the upper part of the water, a body of water like a pond, a lake, stream, and even the ocean. There are specific phytoplanktons. They are just a few terms from ecology. They are the primary producers. If you don't have multicellular plants in the water, they are the primary producers from the ecological term that produce the primary source of glucose through photosynthesis, and also they're very important in the food chain because guess who eats them? The zooplankton come along and they eat the phytoplankton. So these are the primary consumers.
So are phytoplankton microscopic or?
Most of them are.
Okay.
Some you can barely see with the naked eye, like the stuff, the protists that saw in lab. Something else I want to. We focus a lot on what the cell walls are made of, and we also know that a couple other things to remember: gram-positives and gram-negatives. According to the type of thickness of the cell wall, and bacteria have at least some peptidoglycan. Like plant cell walls are made of cellulose. And
You said plant cell?
plant cell walls are made of cellulose. And I'll just kind of do it here too. Fungi are made of chitin cell walls. Which is the same stuff that makes up the exoskeleton of arthropods, the animals, the creepy crawlies with the armor skeleton. Okay, so peptidoglycan, some form of that, and there's a gram-stain reaction that you divide all bacteria into either gram-positive or gram-negative. And it depends upon how thick the cell wall is. Gram-positives will retain the blue stain, and gram-negatives will retain the red. So I would just write that down somewhere so that you know which is which. You always use the same sequence of 4 different stains. At the end, if they're stained blue, it's considered to be gram-positive. If it stains red at the end, then it's considered to be a gram-negative. Like a gram-negative rod. They're very frequent in the colon of the intestinal tract. And that's where E. coli is an example, Escherichia coli. Okay, you want to take a couple deep breaths? I know I'm throwing a lot at you here. You okay? Can't go over anything else? Try to absorb it instead of just writing it down. Try to absorb it as much as you can. So our fungi, in terms of a little bit of ecology, these are your producers. Autotrophic means they make their own food, and it would be by way usually of photosynthesis. They're the producers in the food chain, and then anything that eats other things, these are called heterotrophs. So which one are we?
Heterotroph.
We're heterotrophs because we eat other things. We eat plants, we eat animals. And so we're heterotrophs. And also fungi are heterotrophs. That's why they were taken out of the plant kingdom, because the plant kingdom is all trophic. Photosynthesis, they make their own food. And they have those powerful enzymes that sort of digest. Also, in terms of decomposers that help to return dead material in something that's decaying and to get the nutrients out of it, return it to the soil. We usually think that there are two main groups who help with decomposing things, and that would be bacteria and fungi, right? Bacteria and fungi are our main decomposers. Some other terms associated with fungi, remember, in terms of the structures, hyphae, mycelium, and you may remember from lab that those terms came by, that they are heterotrophs. They secrete digestive enzymes, liquefy the material they're acting on, and then soak it up like a straw. Okay. This chalk is writing really well, but it doesn't want to erase. I won't have to do exercises today. So. Wow. Okay. I hate to say this, but do you remember this section? There are evolution questions on there as well. Now, I know that was a dreadful test. It was one of those, that first test that I used the test bank. And so we're just gonna go over a little bit of the main points of evolution. Now, before I do that, let me tell you that other than stuff like autotrophic, heterotrophic, decomposers, food chain stuff, primary consumers, primary producers, I did not put ecology on it. So that's one of the sections. Other than that stuff, I did not put any heavy-duty ecology questions on the test. Or it would have been much longer than other questions. All right, so, and first of all, what does this word mean? If you can say 3 words, it's a process, right?
Over time.
Change over time. Very good. Change over time. What are some of the evidences of evolution? We may not name them all, but what are some of them? Yes.
Molecular DNA.
Very good. Comparison of the DNA and helping to form phylogenetic charts. What are some other evidences of evolution and change over time? Don't all find at once. I think it said biogeography, finding similar types of organisms, similar types of climates around the world.
Mhm.
Also, fossils,
So
of course, are also a big part of that. Homology, similar origin and structure, even though they may have different functions. If you look at a lot of vertebrates, the whole pectoral girdle and the pelvic girdle are similar, but even in like a manatee or even in a whale or a porpoise that's also a mammal, but it's in the water, they're used more for swimming than they are for walking, but they have a similar structure and origin. So these are homologous structures. And there are some others we could put down, but that should give us a good start for that. When you think of people, that the main father of evolution is who? Darwin, yes. So Charles Darwin, you don't need to know Charles, but okay, Darwin. And he made some observations, so forth, and then he drew some conclusions. He noticed that there are always variations. Organisms differed, like we all differ, hair color, skin color, hair color, eye color, all kinds of differences. He observed there are variations. He said there's a tendency to over-reproduce. But he also said that that is because there'll be a lot that won't make it, or they'll be eaten. But there's a tendency to do all this. What he really said is that he believed that organisms, which are any living things, descended with modification from common ancestors. And the actual main not the only, but the main mechanism by which evolution occurs is natural selection. But descent with modification goes with this, okay? And yes, you could say it's like survival of the fittest. Those that reproduce most. Are the most fit. So they reproduce the most of the most fit. Okay, for that particular environment. We did mention a Hardy-Weinberg. Constant. I'm not going to give you an equation or ask you to do an equation. It's just like concepts. But Hardy-Weinberg equilibrium constant says you will have no evolution, which means changes. Oh, the big one I forgot to mention. What level? Okay, cells, tissues, organs, systems, individual, organism, population, community, and it goes on. But in this hierarchy of living things, which level does evolution work on? Now, you probably want to say individual, right? But don't. Evolution, now natural selection may work on the individual, but true evolution happens at the population level, which is a group of individuals of the same kind living in a certain area together. So evolution works at the population level. Now, another factor, let me finish this. Hardy-Weinberg equilibrium constant says you have no evolution if there's a large population, random mating, in other words, they just mate with whomever, there's no preference, there's no sexual selection. Large population, random mating, no natural selection, I'm just gonna put NS for natural selection. No natural selection, no gene flow, and I think there's another one, large population. I think there probably is, but this should probably help you. At least these four. If these things are held to be constant, then there should be no evolution. The possibility of this happening like this is almost nil, particularly the no gene flow. That would be like migration. You moved from Milford to Batavia. That's migration. And populations do this a lot of times. Now, in terms of you can have something called genetic drift, which usually occurs in small populations. And there are two types. And one is called the founder effect. And one is called the bottleneck. And that is, like the bottleneck would be, there's some catastrophic thing that happens. And there's just a few individuals that are left. Those are left are the ones that can reproduce, and they're more likely to carry on their characteristics when meiosis occurs. Remember, meiosis is the cellular process that shuffles DNA and contributes to great, meiosis 1 especially, to great genetic variation of individuals. So that's more of a bottleneck. Here, founder effect would be that you have a group that decides they're going to leave their population and they're going to move somewhere else, and then they're going to have individuals just with their characteristics. And so there are a couple different examples of genetic drift. The drifting away from the main population. You know, those phylogenetic trees, sister taxa, I would put that down. Sister taxa, what they do is they share common ancestors on a phylogenetic tree, and they show patterns of descent. Okay. Are we good with evolution? Botany. study of plants. We had the lower plants, which are the mosses and the liverworts. They were called the bryophytes. There's also such a thing in plants called the alternation of generations.
Yeah. Fight and sporophyte.
There's a gametophyte generation, which is a haploid, which has half the number of cells.
Like
Haploid. And the sporophyte generation is the 2N
Diploid.
diploid. Now, these lower plants, if you walk up to a moss and you look at it and you say, that's a moss, that's called the dominant generation. Which generation is the dominant generation with these lower plants? Yes. Yes, very good. Gametophyte is the dominant. Okay, so you have the lower plants, mosses and ferns. Gametophyte is the dominant.
So the gametophyte's always the dominant?
It means, don't think of dominant in terms of genetics. It means that the phase in which it's found, you recognize it as a moss. But you see that little stalk that comes up sometimes with a little thing on top? That's really the sporophyte. But to actually recognize this carpet-like growth as moss, that's the dominant generation, and that's the gametophyte, the haploid. Whereas, I'm going to jump from here to here then, the next in line plants are the ferns. Okay, these reproduce with spores, and they need water for fertilization. And these also reproduce with spores, and they also need water for fertilization. By the way, what are those little brown spots called on the back of the fern? Labs are coming back. Sori or sorus. And they've got spores. Little brown spots on the leaves. The fern leaves are called fronds. What are those little tiny, young, unfurled ferns called? That look like the end of a fiddle. They're called fiddleheads. Okay, so fiddleheads are the young, unfurled ferns. These have a dominant sporophyte. If you walk up to a fern, you say, oh, that's a fern. It's a sporophyte generation, the 2N. Now, then there's the big group. And, okay, ferns have vascular tissue, but they still have spores from reproduction. And then you get the vascular seed. Remember, the development of the seed was probably the most important development for land plants, and they no longer have to have water to reproduce, and they have the embryo inside of a protected seed coat and with food, and you have two main groups.
No.
We told you what gymno means, naked. So these are like the pine trees. They have the cones usually, for reproduction, whereas angiosperms are the flowering plants, and there are two main groups. Monocots and dicots. Okay, now in plant, in terms of organs, you have roots. In all of them, you have root stems and leaves, and in these, you also have, well, actually cones, kind of, but the flower is a reproductive part of angiosperms. If you eat a root, a stem, or a leaf, biologically, what is that called? It's a type of vegetable because it's the vegetative part of the plant, which has nothing to do with reproduction. But if you have a ripened, mature ovary,
It's a fruit.
which comes originally from the flower, and the ovary ripens and matures, is called a
Fruit.
fruit. So it has changed our opinion of some things like cucumbers, because oftentimes they have seeds. So a cucumber really, biologically, is that a fruit or a vegetable?
A fruit.
It's a fruit because it came from a flower and then it grew this ripened mature ovary. So the difference between a vegetable would be eating any of these root stems or leaves, and a fruit is a ripened mature ovary. Now, it also goes into the test. There are a couple questions on there about how to tell the difference between a monocot and a dicot. I would look at root systems. And I didn't go into a lot of detail, but also the vein pattern in leaves. If they're parallel veins, are they monocots or dicots? Like corn, and like a lily. Those are monocots. If you, and then the root system is a fibrous, like grass that is in our lawn. That's a monocot. And it has a fibrous root system which helps to hold the ground. Whereas dicots have something more like a carrot,
A taproot.
a taproot for dicots. But remember the flower part numbers. Which one's which? Yes,
Monocot is 3 and dicot is 4 or 5.
excellent, yes. So monocots are flower parts in 3s, multiple, and these are 4 and 5s. Okay, if it has 10 petals, it would be a dicot. If it has 6, it would be
Monocot.
a monocot, because 3 times 2 is 6. Yes.
I felt like 12.
Yeah, then you look at the leaves. If it has more of a palmate pattern or more like a feather pattern of the veins in the leaves, then it's a dicot. Or you also look at the root system. You're right. That's a hard one there.
It's a tricky.
The next would be zoology, and then it goes into human anatomy, physiology. I think I've only got about transpiration also, since we're in botany, is the evaporation of water from the leaves, which is sort of like perspiration in people. It's transpiration, loss of water. Stomates are the opening in leaves through which the gases are exchanged. Vascular tissue, that's the other thing for botany. Xylem and phloem. Which one's food, which one's water? The X is near the end of the alphabet, which matches the W.
Water.
So xylem is water, and food flows through the phloem. Okay, so food flows through the phloem tubes, and those are the vascular tissues. Okay, so there are a few zoology questions, and then there are several more of those anatomy physiology. I'd probably reread through that anatomy physiology test. Yes.
Are you gonna accept test questions for the?
No, there are no test questions. And what the syllabus says, there are no extra credit points other than notebooks during the last week. So, and then, of course, I won't see you again after. So be here. That's the important part. Your final exam is in here, in this room, on Monday from 9:45 to 11:45. It's in the syllabus. Monday, UC sets it. Okay, so 9:45 to 11:45 right here. Be sure that you show up to take it, because you don't want to zero out of 100. Bring your notebooks, that can help you. Thank you very much. Have a great day. Enjoy your weekend study.
But